17 talks about memory and learning, and it's segment 4 starts talking about is LTP a mechanism for memory formation, and starts with talking about correlational observations, and its time course of LTP bears strong similarity to time course of memory formation, with memory LTP can be induced within seconds, may last for days or weeks. So that's like short-term memory. Somatic intervention experiments show like pharmacological treatments interfere with basic physiological processes that contribute to LTP to impair learning. Behavioral intervention experiments are the most convincing evidence between LTP and learning and behavioral LTP. They demonstrate that training an animal in a memory task can induce LTP in the brain, and it's difficult to see where to place electrodes to show this actual growth. But it's also like fear conditioning as an example, and LTP can be a kind of plasticity almost. Some simple learning in mammals relies on circuits in the cerebellum. Although LTP might be the synaptic mechanism of memory processes require an analysis of neural networks. Difficult to describe in humans because of the billions of neurons that we have that are not in fixed circuits, and that they focused on around classical conditioning. So associative learning is learning of relation between events, a stimulus and response, or a response and its consequences. So it's different than habituation and other learning because it occurs later in life. It can occur later in life. Instrumental conditioning, or operant conditioning, is an association formed between animal behavior and the consequences of that behavior. And that's by B.F. Skinner in the Skinner box, where the rat would press on the bar and receive a food reward until it learned that pressing on the bar meant food. So then there's classical conditioning, which is initially a neutral stimulus comes to predict an event. So that's Pavlov's dogs, where the unconditioned stimulus, that was the salivation, when he saw the food, which was the unconditioned stimulus, which was when he saw the food, and the unconditioned response, which is when he would salivate, was coupled with the conditioned stimulus, which was the bell, and then obviously the conditioned response was the salivation in response to the bell. So this can also be accomplished with rabbits, and it's a puff of the air to the eye that causes blinking. But if coupled with an acoustic tone, coupled with an acoustic tone, coupled with an acoustic tone, the rabbit will actually learn to blink when the tone goes off rather than just when the actual air touches it. And so this is how this works is there's sensory fibers in the cornea that run along the trigeminal nerve to the nucleus in the brainstem, where the interneurons send axons to synapse on motor neurons that activate the eye muscles. And the hippocampus is not required for this type of conditioning, so they kind of just bypass that. So on to complex eye blink conditioning, the trigeminal pathway that carries info about the corneal stimulation to the cranial motor nuclei also sends axons to the brainstem in the inferior olive. These sends axons called climbing fibers down to cerebellar neurons in the interspasticitous nucleus. Local cooling or drugs that block GABA receptors have the effect of reversibly shutting down the interspasticitous nucleus, and if performed at the beginning of the training, then no conditioning occurs until after the effect wears off, and vice versa, whereas if it's given after the training's already occurred, it won't affect the training. Researchers have been able to show that LTP probably plays a role in human eye blink condition, and that leg flexion is cerebellum dependent. In the adult brain, newly born neurons may aid learning. So neurogenesis. Adult neurogenesis occurs primarily in the dentate gyrus of the hippocampal formation, and new dentate neurons that survive and grow ultimately receives inputs from the entorhinal cortex via the perforant pathway. Therefore, new neurons appear to integrate into the functional circuitry of the hippocampus. An adjacent cortex. Neurogenesis in experiments can be increased by exercise, experience in an enriched environment, and memory task training. Reproductive hormones and experiences also potently influence neurogenesis. Neurogenesis in memory may be limited to the hippocampus-dependent learning, such as spatial memory and fear conditioning. So it talks about a conditional knockout, which is a gene that's selectively deactivated in adulthood in specific tissues that resulted in marked impairment in spatial learning with little effect on behavior. So it only affected their learning. This impaired learning that accompanies reduced adult neurogenesis and is consistent with the role for new neurons in learning. Animals with increased survival of newly generated neurons resulted in an improved performance, enhanced hippocampal LTP. Learning and memory change as we age, obviously. So age-related memory is hard because motivation, early education, and other confounding variables affect it. Aging people have a decrease in their ability to complete tasks that show conscious recollection and that rely primarily on internal generation of the memory rather than external cues. So rather than something being brought up to you by someone else or you seeing something that brings that up, actual just recollection is hard. If only vocabulary, older adults actually outperform younger adults. Aging causes decrease in spatial memory and navigational skills. In severe cases, elder amnesia can result in someone to mimic an infant. Age-related impairments of memory have several causes. Impairments of encoding and retrieval. Older subjects show less cortical activation than younger subjects when encoding or retrieving is self-initiated. Less frontal and temporal activity is seen in older subjects while learning new faces. Still shows comparable brain activity to younger subjects, though. So despite the fact that their brains still function in a similar way, they just actually don't have the frontal and temporal activity, but the rest of the brain is similar. Loss of neurons and or neural connections. Brain gradually loses weight after the age of 30. And some parts of the brain, such as the frontal cortex, lose volume or weight and then lose more than other parts. Problems with the cholinergic neurotransmission, so acetylcholine and things like that. Two subcortical regions known as the septal complex and the nucleus basalis of the minor provide profuse cholinergic inputs to the hippocampus and cortex. And this is seen in Alzheimer's. Drugs that improve acetylcholine improve memory performance. Can the effect of aging on memory be prevented or alleviated? Which is obviously a very common thing because people want to be able to be smart and not lose their memory for as long as they can. So there's these things called nootropics, which are drugs that enhance cognitive functions. And so the use of drugs such as donepezil that inhibit cholinesterase, resulting in an increase in cholinergic transmission in the forebrain, has a positive effect on memory and cognition in mild to moderate cases of Alzheimer's. So rather than inhibiting the uptake of cholinesterase, lets it sit there for longer and affect you more. Compounds called ampicans act on glutamate receptors to improve hippocampal LTP, raising the possibility that compounds that alter this kinase could be highly selective memory drugs. Rats raised in enriched conditions showed improved handling of stress and associated reduction in glucocorticoid secretion and lower chronic levels of glucocorticoids. Enriched experience also increases the release of nerve growth factor. Lifestyle factors include living in favorable environmental circumstances, involvement in a complex and intellectually stimulating activities, and having a spouse or partner with high cognitive status. Thank you.